Very pretty picture for anybody. All right, so anyway, this is the first class after the midterm. And I'm going to show you where a few things are and discuss a few things about what's here. First of all, after the curve, and I'm not even going to tell you what the curve was, but after the curve, 7 people made an A in here and 4 of them got 100%. Most of that 100% came from the people who absolutely kicked butt on the word questions, because there was 80 possible points on the word question.
80? Oh,
80.
One of them is 2.
Yes, 2 of them.
2 of them.
So that was great. A huge number of people, which means you know I'm going to do it again, did not read the directions. And on the directions of the exam, it says, if you read these directions, record the answer to number 206 as mycobacteria. 10 points free, 20 multiple choice questions. If you don't learn to read the instructions, I am going to keep doing it. You know, if it says when you're turning on some sort of respirator at the hospital and you don't read the instructions and it's not the model you were trained on and you set it up wrong, somebody's going to die. Read the instructions, always. So anyway, in your class, 7 people make A's, 2 people made B's, 6 people made C's, 5 people made D's, 6 people made official F's because they came here and they actually flunked it. But a total of 15 people got an F. And that counts the 5 people that dropped and then the other people that just didn't bother to call me or show up. Of which 4 had a B, which still upsets me. Which, if you look at the night classes, the other night class, it pretty favorable comparison, similar, actually better than usual for my midterm. My midterm usually averages 67. So
What was the average?
I haven't gotten it completed yet because I want to do these 2. I was up till 4 last night trying to get it all done. Anyway, so that means that everybody's going to be getting a midterm update grade sheet, and there will be another set of bonus questions available for everybody in the class if they have them done by the 22nd. So we're going to talk about that. Friday is how to type a perfect unknown report, and yes, I'm going to go a little bit over it next, but if you want to know all the details and you want to look at a couple of them that I don't have online, you can certainly come, and I will be talking about that, and it would be very nice if you have questions. So you should look into how to do it before you show up. So if you have questions, then we will be able to answer them for you. And remember, there is a couple of things that I want to make sure people know. Whatever data you got is right. You cannot collect data that's wrong. So if you put what you got in your table, your data table, it cannot be wrong. If you lie, it's not only an F on it, it's a 0 on a required component of the class. What is lying or cheating on the paper? Saying we did a test we didn't do. That means you copied a paper from a previous term. Every term we do slightly different tests. So if you put in there lysine iron or peptone iron deeps, I'll know you either didn't know squat or you lied, both of which are 0s, because we didn't do that this term. If you say blah 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 test, I got a positive, remember there was one test that didn't work. I'm the only one that knows it. So if you got a positive, there's going to be some serious repercussions because it didn't work. The reagent was bad. So whatever you got is perfect. All right, when you go to the data books to fill out, remember, you're supposed to go through 6, 7, 8, 9, plus systematic and McFadden, or anything online, and find something that most closely matches your results. So you're going to have a data table, and the first column is going to be every test we did, alphabetized, which is the same first column of the methods page. Then there's going to be a column, and this is going to be called tests, and the next column is going to be called you. Now don't put my name. Every term I have somebody put Hicks, or if I change it to my name, they put my name. Put your last name, Smith, McGillicuddy, Nguyen, whatever, put it in there. And then you should have a result for each one of the tests. These are the ones you did. If you didn't do that test, don't put a result. Just leave it out. And remember, there are several tests that have more than one result, like Ch10AViroidsPrions. It has whether or not you're gram-negative on there. It has a color, how it grows on agar. If it's clear, it's lactose negative. If it's salmon, it's lactose positive. If it's green, it's Shigella. If it's blue-green, it's Salmonella. Or no, just the opposite. But then, anyway. And then it has hydrogen sulfide crystals. So there are 3 things you should have recorded about Ch10AViroids. Litmus milk. There is no, don't be giving me positive on litmus milk. That's crap. There are 2 things on litmus milk. Lactose and casein. On Kligger's iron, there are 3 things. Glucose, lactose, and hydrogen sulfide, and you could even have a gas on glucose. So don't be telling me positive. That'll drive me nuts, because that means you didn't know squat. Alright, so on the data page, you're going to have reference 1, and of course you're not going to put reference 1, you're going to put like Bertie 6th, and 7th, and 9th, or whatever, as the other columns. Remember, this one most closely matches your results than these. And the newer the book, the less results it quotes. It might have only 5 results, because nowadays they only record in the reference books the results that differentiate it from another genus. Not all the tests like we did. Finally, what if you got something no one else on earth has ever gotten? Like use on motility. And you got a positive, and every reference book you checked said negative. Then you have to put an asterisk in front of motility, and down here you say, this unusual and strange and twisted result. No, you don't have to say twisted or unusual. You can just say, this unexpected result. It's explained in the 3rd paragraph of the conclusion, which we call the excuse paragraph. How I could have possibly done this wrong. Alright, so anyway, these are some of the hints. What do people do that cheat? They add tests we didn't do. They put a list of tests that we did and they make up results. Or they go to a reference book and it doesn't include that, so they just put something. All of which, remember, in 23 years, we've had the same 35 unknowns. I have memorized them all. I know what it should be. Don't lie, cheat, steal, or alter the results. Nothing you do from a reference book and nothing you did on a test can be wrong. All right, yeah?
We have repeated a test. Does that go in this data table? Twice?
Yeah, you could probably put like motility 1 this, motility 2 that, and you can even write inconclusive if you couldn't, and that's why you repeated it. Okay, so anyway, that's a little thing, a little warning about the paper. And let's see if there's anything else. Let me show you where to find some things. And it looks like one of the things I thought we could find is not there, but anyway, everything for this stuff is on Last class material and comprehensive final exam. Oh, final exam dates. May 28th, Tuesday at 1:30. May 29th, 8 a.m. May 30th, Thursday at 5:30. Those are the three. And if you want to go to one that's not your class, and remember your class is Wednesday, May 29th, 8 a.m. If you want to go to one of the other ones, you have to email me and get a seat number. All right, going back to comprehensive final exam material. This tells you a little bit about the final exam. This tells you a little bit about what's in it. And then to get the notes for the lecture, there's the Viroid PowerPoint, and there's the HIV PowerPoint. Where to get the information that that's made from, click on Chapter 11, Eukaryotic Parasites. This tells you about the unit, Eukaryotic Parasites. This tells you the topics, Viroid, satellite prions, then comes general viruses, then comes HIV/AIDS. And then down here, this is a list of the videos that are available. Sometimes we see more than others. And then this is where the notes for today start, where it says acellular or subcellular parasites. And this is viroids, satellites, and prions, which is spongiform encephalopathies. So, and this is important terms that you're going to need to copy into your lab record book front to back under brain eaters. So remind me to put that back up there in just a minute. Actually, so you're going to put, remember all videos are front to back. So you're going to put the brain eaters at the top of the page. And then for each line, you're going to put one of these phrases that's separated by a comma and fill in as you go, listening to the video, and that's the questions. And each one of these will be a question on the bi-weekly test. But going boringly backwards to other things you might need to know, and this is final exam packet, and it looks like it's missing. So if it's missing, remember the other thing you can do is go here and go into the link for the drop. Let's see. One. There we are. Micro website log on. Navigation. And there'll be one over here that says final exam packet. Exam packet. And this tells you what goes in the final exam packet. So I'll have that up soon. Somebody remember to send me an email about it. Oh, good, thank you. All right, so let's go back to this and talk about what we're talking. Oh, let's talk about one thing I want to talk about: how to get the extra 25 points. If you go down to eukaryotic parasites, here it is, eukaryotic parasites. You are to answer these questions about each of the 5 great classes of eukaryotic parasites. The name of the topic is the 5 great neglected diseases of mankind. They're not exactly 5 diseases, they're 5 types of diseases. And you are to answer 7 questions about each one on a card or a sheet of paper, probably easier on a sheet of paper. And luckily for you, I already have it done. You just need to organize it in a nice, clear, matte manner. And here they are. The tissue roundworm diseases, which includes river blindness and heartworms, eyeworms, any worm. Then the non-segmented flatworms, which are the flukes. The greatest disease of mankind, malaria, which is plasmodial disease. It's called the greatest because it's killed over 1 million people for 5,000 years. And for the first, until the last 10 years, it killed 10 million people a year. Yet we get hysterical when 1,100 people get HIV/AIDS, and this is still killing tens of millions. Then the last type of disease is called the trypanosome protist, and this is African and American sleeping sickness. In America it's called Chagas. In Africa it's called African sleeping sickness. And so those are the cards, or let's say sheets of paper here to answer the 7 questions about. So you can get the information here, and please do not cut and paste. Remember, you need to change it into your own words, and you answer these 7 questions about the 5 great neglected diseases of mankind. Why will that help you? Because on the test, you're going to have those questions. So that's why that study sheet is due on that day.
Is this part of the packet? Yeah,
No, the cards are due on the day you take the exam, the bi-weekly exam on chapters 10 and 11, which is
The 22nd.
22nd and 23rd, right? All right, so let's talk about what we're talking about today. And I like to actually do it from here because it shows up a little bigger on the overhead machine. Come on, baby. Do your thing. Love it. Like that. Okay, then we'll go back to the low. Ever since Flash started revising every day, it's become basically useless. You gotta remove this.
Question of the annual report.
Yeah,
So, are we doing the results for the genus or the species?
both.
So it would be, if they're conflicting, then we would have to list it twice on the data table?
No, if they're conflicting, the species wins. Let's try going up. Sorry. She did remind me of something I haven't done, and that was a little bit more on typing your unknown report I'd like to give you. So, how to type your unknown report. All right, so what we use is called modified APA style. And modified APA, American Psychological Association, way back when, decided to put together a publication guide, a little book. At first it was a pamphlet, now it's like this thing. And it tells you how to type a paper, or a newspaper, or a news release, or a thesis, or a dissertation, or for submission to a reputable journal. It tells you the format to type, just like an English paper, but it's different. Remember, an English paper has ibids and footnotes and little numbers up in the air. We don't do that in science. So the American Psychological Association made a manual, and because they made it, we decided to, with just a very few modifications, use it for all of science. So that's why it's called APA style. And our few little pieces of modification is called MLA, but you don't need to worry about that. So key things, once you have it all typed, and then, 10 to 12 pages is about how long they usually get, please just staple a corner. Don't put it in some pretty plastic folder. If you want to see how to torture a teacher, give them 30 plastic folders to carry, because they're like ice, and they run all over the floor, and you can't fold them, you can't hold them. So remember, just staple it. And your unknown report, the very best thing to do is to bring it to the class that you actually are in and take the test. And while you are taking it, I will grade it. Now, what if there is some printer problem, North Korea attacks or something like that? Then you can bring it to one of my other classes. If you don't bring it to a class, it is minus 25% per day. If you come and bring one that's a piece of crap, I will then write 0 on it, redo within 12 hours, and you email it back to me as a Word attachment. All right, so let's talk about what goes on. The title page is going to be different than MLA because you're submitting this for a class project. And so on the title page, you're going to have the name of your unknown. And who was it that hadn't confirmed theirs? What was the number?
28.
28. In this class, right? 0931? Oh, 30? 31? 31. 0931. And what was that number again?
28.
28. And what did you get? You got Shigella, right?
Yes.
What was your species? Sony or Flexneri?
Flexneri.
It was Sony. There's only one difference. Yeah.
I sent you the email about yesterday. I was on Shigella, number 25.
What number did you have?
Number 25.
25 in this class.
And you said, I asked.
Flexneri, yeah. Paradysentery or Flexneri. It goes by 2 names. So in the 6th edition and before, it was known as Flexneri. Then in the middle of the 6th edition publication, they changed it to Paradysentery. So you're going to have to look up both. Yes?
I have Klebsiella, number 19, but I don't have this species.
Pneumonia. Yeah,
Number 18.
I like the word alter.
Enterobacter, and I have Aerogenus.
right. Anybody else not sure about their species?
I have 26, Shigella, and I Flex.
Yep.
Flexneri, yeah.
Or Paradysentery?
How is
Okay.
it possible that there's different numbers in every scheme?
Different class, different numbers.
I have Shigella Flexneri also.
Right,
And I'm 25.
I know.
Those are just, it's the same thing as.
I have 35, and I try not to give gram positives because you can run 3 tests and know what your gram positive is. And so I usually give the same bacteria with different numbers to 3 different people. All right, so anyway, on the title page, it should start out with unknown number 36 was identified as Shigella Flexneri, or Flexneri was identified as unknown 26. Whatever it is, that's your title. And remember, genus and species is always underlined. Genus is capitalized. Species is not even in a title. And then below that, you could say by, put your last name, then your first name, your ID number, your class number, your section number, the date, your email address, and your telephone. Why? If it's late, if somebody slid it under my door, or some other disaster occurs, I want to be able to call you and say, what the hell is this? Or, I don't understand you left out page 6. Where's the data chart? Email it to me, quick! Alright, so that's why I have email and phone number on there. Don't put your mama's phone number unless you want me to call your mama. Alright,
It says on the blog, on the page, for the paper, it says links below for the online.
Yeah.
rankings. It's not, it's also.
okay. Well, you can go the other side. The only online edition of Bergey's is the 7th. 6th, I gave you a copy of if you're gram negative. If you're gram positive, I have a copy of the 6th. 7th is online, 8th, I have 3 copies, and there's 2 in the library. 9th is everywhere, it's like, you know, garbage, it's everywhere. Every library has it because all librarians throw away all the old editions and keep the latest one. But I have a copy and the library has a copy. So this is 6th, this is 8th, this is 7th, and that is 9th. And then I no longer have a copy of McFadden 3rd edition. Someone stole it, never returned it. So I think it's in the library. You can also, for one of your minor, a lot of books have actually been written about certain bacteria. So you could find a book that has a table with at least 4 or 5 of your results, and that would be fun. All right, the second, that's the title page. The second is the abstract. The third is the introduction. The fourth is a methods table. The fifth is data table. Then there is the conclusion and the reference page. And usually the best way to talk about something is to actually show you a copy of it. And so I will try to go back here and see if I can pull up a copy of it. Okay, the parasite lecture that I'm going to give you in a few minutes is right here, but right now we're going to look at the unknown paper. And if you go to unknown paper and then go down to see an example paper, this is a pretty good paper here. This is the second one is the better one. So I'm going to show you, see which one is the one that's better. All right, so here's what the title page looks like. Unknown 7, Klebsiella pneumoniae by last name, college, ID, class, phone, email. By the way, where it says page break, do not copy and paste this. I actually have people that just do a complete paste and paste that into their paper. Okay, the abstract. I know, some people just don't care. All right, there are 2 ways to type the abstract. If it's on a page by itself, you have to have the title abstract. And it is squared exactly in the middle of the page, and it's typed single or double spaced. We prefer single. And you just type it there and put the references in it. If it's typed as a part of the paper, and you can do it either way, then you don't put the title of the paper, you just put this abstract indented, squared, justified on both sides, just like that, and then the introduction begins below it. And the introduction doesn't have the title introduction. That's what the parentheses mean, because it's beginning, stupid. So everything in science on these papers is to cut down verbiage and stuff like that. We want as little as possible, no crap. So it'll be totally different than your English paper. All right, and how to type it, you don't put the word abstract up here if you're putting it on the same page as the introduction. All right, let's talk about what goes in the abstract. It's very simple to write it. It's very modular. You say, unknown number 36 was received on April the 1st. I used a four-stripe quadrant street isolation method to isolate my unknown from Staph aureus, the known. After doing the Doxorus and purity test, I determined my unknown was a gram-negative rock, or whatever it is, of course, period. Then you put, I performed the following test on it, colon. And then you list every test you did with the results in parentheses. It is the world's longest run-on sentence. English teachers have been known to fall over and froth at the mouth. We don't care. It's not English. All right? And then once you list all your results, and remember, don't include anything you didn't do, then your last sentence is going to say, using the dichotomous key, and then you're going to put my website or blog site in it as a reference. And then you pick the Bergey's manual closest to your results or whatever. Bergey's 6th edition, I determined my unknown was, that unknown number 7 was, and then you write the name and underline it. So it's quite easy to write an abstract. People freak out about abstract. It's very easy. What's the purpose of an abstract? Anybody know?
So you don't have to read the whole article.
Right. They publish books of abstracts about microbiology, about anything. And what it does is it gives you a hint on what they did, a hint on their results, and then if you want to request the entire article, you can. That's the purpose. And so that's how to do an abstract. Now let's talk about the introduction. And the introduction is the only place that you can do BS. And the introduction is always typed the same way. It's typed double-spaced. And it does not have the title introduction. And when you have a reference, you put a parentheses, like you were referencing my blog site, you would do Hicks 2013, closed period. So that's how you do references. You do the author's last name and the year published for everything. And we'll talk about on the reference page. Then on the reference page, you put the authors alphabetically, and so you would put Hicks, Donald R. Then you would put the not title microblog site 2013 or whatever. Then you would put the address. Then you would put accessed on a certain date. And then you would list the pages if it was a book. So if you looked at page 460 first, and then you quoted page 13, that's how you would do it.
And if it's the website?
If it's a website, there is each page has a different specific heading up there, but you can just say the whole website and just say, but you do have to say accessed on the 23rd of May 2013 because websites change by the moment at 8 a.m. All right, so anyway, what do you put in the introduction? It's easier to tell you what not to put in. Don't put anything about your unknown. The introduction is why we do unknowns, how we do unknowns, and cautions to doing unknowns. So why do we do it? To replicate what happens in real life in a hospital situation when someone comes in with a bacterial infection. We're trying to identify it so we can treat it. And then you might talk about streak isolation, and the reason we do that is Koch's postulates allows us to isolate a microbe and determine what diseases it caused, and then you can quote Koch's postulate. Then you could say something about maintaining log growth, because all references use log growth, and you do it by incubating overnight at optimum temperature and maintaining it in the refrigerator for 10 to 14 days and subculturing after that. And you might want to put something in there about maintaining sterility and aseptic techniques, so you didn't identify something from the environment and not your original unknown. So it is a general quote of why we're doing this and the rules for doing an unknown. And you can read the one that's there and get an idea of what to put in there. All right, after the introduction comes the methods page. And the methods page is a table. Now, legally, you can type it 2 different ways. Please don't type it the second way or I will kill you. The legal second way is back when they didn't have table makers and tables were a nightmare. I remember my first table on an Apple II where I had to type in what I thought, print it out, and if it was office space, go back and hit back because it wouldn't, it didn't center things for you or anything. It was a nightmare. So legally, you can type the methods page as a story, a prose story. On the 23rd of April, I received my unknown. My unknown was number 27. I took a loop, I heated it to red hot and cooled it. I dipped it once into my unknown, and I took a freshly poured, cooled auger plate and lifted the lid and streaked 4 parallel stripes. I closed, you see the story? Yes, someone did it once just to irritate me. It was 45 pages for the introduction. After the 3rd page, I wrote, crap! I'm not reading any more of this on it. So, please just do it as a table. And remember, all programs now have a table maker. Oh, and tables have to be on pages by themselves. You can't have the type run into a table and then put the table in and then start some other section afterward. They have to be removable by themselves. All right, so a table, the name of this one is methods. The first column is the test you ran in alphabetical order. Second column is where you got how to do it. Now, where do you get this? It's from the blog site, and it gives you, at the top of each page, there is a unique address. It always starts with the address of the website, and then it has a slash, and if you look at this one, it's faculty, LA City College -edu. That's the main website, then slash this page, HicksDR, then this page is unknowncoli.htm. So you put asterisk unknowncoli.htm. And asterisk at the bottom, you put the name of the whole main website. So every page has a unique answer here, and because people copied these, I didn't put them in. You have to go look each one up, it's not hard. All right, then if you didn't do exactly as your cards say, you have to put the exceptions. Remember, science means that everybody can copy exactly what you did, do exactly what you did, and get exactly the same results you did. So if you read it after 15 days on the VP and it said to read it at 10 to 14, then you have to put an exception. Anyone not understand the methods table? It's just busy work. All right, the data table, and remember you put the name of the website where you got it down at the bottom, and then the unique identifier that comes after HicksDR goes here. All right, on the next page is the table, the data table, the most important part of the whole paper. And you're going to have, let's see, I think 5 columns. First column is the same first column as the methods page, all the tests you did. The second column is your results. Put your name, not Mark Insco, not mine, yours. Bloom, right? Broom, something like that. There. All right? And then put your results in here. Now remember, if your results are different than any reference ever found, you're going to have to put an asterisk in front of that. See it there, he has a urea result that no one's ever gotten on Earth before. So you have to put a asterisk in front of it and say this will be explained in the third paragraph of the conclusion. So you put your results, and then you put the reference that most matches yours, and notice there'll be lots of blanks because they don't do all of them. Then you put 2 other references that have at least 4 results in them, and the data page is done. At the bottom of the data page, you need to put a key to symbols used. Believe it or not, plus does not mean the same thing in any book. In some books, it means 90 to 100% are positive. In other books, it means 100% are positive. So if you use a symbol, then that symbol needs to be explained somewhere down here. And also, let's see, you will need a key to symbols. And then also, if it has an asterisk, this unusual result will be discussed, it's supposed to say in the third paragraph of the conclusion. All right, so that's the data table. Now the conclusion. And the conclusion is it's the only part of the whole paper that's not busy work. I mean, all that was just busy work, going and collecting the stuff and plotting it in a table, and then typing. This does require some thought, so 2 of you are going to get it right. No, I'm just kidding. All right, the first paragraph is easy. Any idiot, in fact, my dog wrote this one. All you do is take that dichotomous key and put it in words. A gram-positive cocci that was catalase negative, casein negative, was found to be the genus Streptococcus. Then you reference the website. That's the first paragraph. A word story of this. Second paragraph is the hard one. How did you determine your species? When you look up that genus in the book that most closely matches your results, there will be 3 or 4 species. You have to make a table of each one, and usually it'll be only 1 or 2 things. Like mine, I think Shigella was one where the motility makes a difference. So in mine, Shigella, the 2 main species I was told to pick between, my results indicated a positive motility and it agreed with the reference, while the other one was negative. So motility was the main test that I used to determine my species, which was confirmed by the professor. Then you put the reference of the book where you got that. This is the only difficult paragraph in the whole book. Do not list every test you perform. That's what people used to do. They used to list every test they did. Just copy the abstract, cut and paste. This requires an actual doing something. All right, third paragraph is what we call funky results. And people used to type, my funky results were. So I don't say funky results anymore, because the word funky should never be in a publication. You say, my unusual and unexpected results included the urea, where I found it was positive and no reference shows it's positive. So that I can conclude that either I did the test wrong, the reagents were bad, it inoculated it wrong, or it became contaminated. So remember, if you got a positive, and it should have been negative, most likely it was contaminated by the air. I mean, dead things don't rise up and become good. If you got a negative, and it should have been positive, there are lots of reasons. Your unknown was dead, your loop was too hot, the reagents were bad, you did it wrong, or you read it wrong. Okay, so that's the third paragraph. And then the fourth and subsequent paragraphs are interesting and amusing information about your unknown. Things like who named it, what year, where it's found in nature, and remember, in this has to be one thing: how your unknown relates similarly to a pathogen. Remember, we don't give you pathogens. So if you had Shigella, then you should relate it. My unknown was given to me because it resembles a deadly food poisoning called Shigella dysentery. So you need to relate your unknown to something in real world life that causes a problem, that is in the same genus. And then you write, you know, it's normally found in the soil, in somebody's poop, or, you know, in a chicken's cloacas or whatever. So that's what and you get that from either Googling it or just looking in your reference book. It says who named it and what year. Yeah. Cloacas is what the egg comes out of. Yeah, they want to get more specific. All right, last part of the paper is references. References are typed really weird in science. First of all, there is no numbers out here. They are alphabetized by authors or editors' last name, unless there's more than 3. And Burgess has 27 editors. So you wouldn't want to use that. You would just use the title of the book. Burgess' Manual of Determinative Bacteriology. Notice, the only thing capitalized in the title is the first word. The rest of it's not. Then you put the edition, the date published, where published, and then you use MLA, copy the page numbers in order that you quoted them. 646, page 13, page 230. That would be the 3 times you quoted that reference. Notice that they're typed single space in a reference, double space between. Notice that second and third lines are indented 5 spaces. And how to do a website reference. If you're not using a book and you're using a website, let's see if he used a website. Ah, there it is. Okay, so you put the name of the article, you put when it was received, you actually should put the time also, and the address to go get it. So that's a general idea of what to do. I'm going to have some copies of an old paper for you that's pretty good for you guys to look at on that day. If you can't come, we'll have it recorded, and you can see it from that. What's your one, little pump?
We have it on private stuff.
Yes, I'm going to go, you know, I'm going to take questions, and I'm going to have a handout for everybody of a fairly good paper so you can see it and take it home and touch it.
What site were you on?
Huh? I was on how to type your unknown paper, and then it said example unknown paper at the bottom. See an example paper? Okay, this is on the old website. All right, so let's talk about today. Other questions?
Yeah, are you cool with using the first person in the paper?
I everywhere?
I mean,
I don't know, read that one, see what it looks like. I don't know.
it's normally, I mean,
I think
you
most
don't
people do it
usually,
in third.
I just, because you kept saying I, so I just didn't know if that was okay.
I don't know.
Okay.
Look at it and see. I forgot. My brain's kind of fried. I had 2 hours sleep. All right, so we're going to be talking about acellular or subcellular parasites today. And we're going to talk about the smallest thing that causes a disturbance in human health. Test question. Viroid. Viroids are the smallest thing that causes a disturbance in human health or plant health or living things health. And a viroid is a naked piece of RNA. It is low molecular weight, usually single-stranded RNA. It looks something like this. It has a few double-stranded regions. So this is the double-stranded region, and these are the single-stranded regions. And it is not in a protein coat, not surrounded by an envelope, and remember, these are not cells. They are 100,000 times smaller than cells. They can only be seen with the most powerful electron microscope. They are viroids. They are subviral, even smaller than viruses, just RNA. And all of them, except for one, so far, are animal disease agents. And they are major problems with crops. What can you do about a viroid? Nothing. They burn the field, they plow it under, and you cannot plant the same thing for 25 years. Now think about an avocado orchard viroid. That means an orchard that took 15 years to get into production of the trees. If it gets a viroid, your entire orchard is gone. You have to cut them down, burn it, and plow it under. And you can't plant avocados there for 25 years. So these are serious diseases of food products and farm products. There is one animal viroid. It's called hepatitis D, or delta hepatitis. And remember, the word hepatitis just means infection of the liver. It doesn't mean it's a virus or a viroid or an RNA virus or a DNA virus or whatever. It just means infection of the liver. So there is one infection of the liver that is caused by a viroid, confirmed. It's called delta hepatitis or hepatitis D. Some people call it dead, because if you get it, you're dead. What's unusual about hepatitis D is that you have to be co-infected with a current infection of hepatitis B at the same time, because one of the enzymes of hepatitis B is required for the replication of hepatitis D. So the good news is, if you have had hepatitis B, you are immune to it. If you've had the vaccine, you are also immune to it. But remember, hepatitis B vaccine requires a booster every 4 to 5 years. So you may have had it. Remember, you went in and took it. You came back in 3 weeks and took it again. Excuse me. And then 3 weeks after that, you took it again. And then they took a blood sample and did a titer to see if you had enough protection. And lots of times you even get another one. And that whole process wears out in 4 years. So every 4 years, you should have a blood sample to see if you're still immune to hepatitis B. Remember, if you have taken the vaccine for B, you can't get D, because you have to have an active infection of B to get D. So basically, there is a vaccine for both. Hepatitis B vaccine will protect you for both B and D. Most people don't think that viroids have ever touched them, but if you look out at those very tall coconut palms or palm trees near USC, those are native to Southern California, and they will all be dead. They are all infected with a yellow palm viroid. If you go to South Florida, you will not see Floridian palms that are native to Florida. They're all dead. Why? Yellow palm disease, which is a viroid. So how do you get viroid diseases? Contact. A tractor rolling through the field that crushes some of the stems and gets the liquid on their tires and rolls through an uninfected field will carry it. You can walk through a field, step on the plants, get it on the bottom of your shoe, and walk through another one and step through it, and you get it. You can transfer it. How did all the palm trees in Southern California get it? Nobody has a 200-foot lift truck to trim the palm trees. So you call the city uses a chainsaw, and if they don't sterilize the chainsaw, which they don't, between each tree, then every tree that's trimmed by the city gets infected, and they will all die. And there is no cure. So what are we going to do about palm trees in Southern California? Southern California without palm trees would be weird, wouldn't it? So they import Jamaican hybrids or Saudi Arabian palm trees that look completely different and are squat, shorter. And they are immune to yellow palm viroid disease. But unless somebody does electroporation and makes a California palm tree that's immune to yellow viroid, there won't be any more by the time your children get up. Okay, just to let you look at some of the viroid diseases, I have a link here, and you can go and look at, and it will actually tell you, hopefully, the link is still good, good. It tells you all the viroids, and they are now numbered, and you can see the length of RNA bases that it is, and you can see the different fruits that are destroyed by viroids. Apples, grapes, chrysanthemums, citrus, coconut palms, coleus flowers, columnia, I don't even know how to say that, eggplant, hops, which really breaks my heart. That's what they make beer out of. Papita, Mexican, mulberry, peach, pepper, potato, rice, rubber, tomato. All of these are infected with viroids. And remember, if you get a viroid, that's it for that field for 25 years. Any questions about viroids? That's all we know. Yeah?
How do you know if your plant has a viroid?
Well, usually what it does is it studs, it stops growing and the leaves shrivel up. Sort of like if you didn't water them, but you are.
Right.
And usually, like I said, it's crops and a few flowers. And by the way, they use the flowers to get very strange flower. Ever seen that striped tulip? It's a viroid. They use a viroid to get a very strange, beautiful flower. The problem is the stem is extremely weak, so you'll see they're taped to sticks to hold them up. But they actually use a viroid to get a certain look on the tulip. All right, satellites are two viruses circling each other. They are unrelated to each other. They're always associated, but they are unrelated. In other words, they didn't evolve together. They just happen to be together. And you'll notice that there's only a few DNA satellites. Most of them are RNA satellites. And there's two different kinds of satellites. There's satellite viruses and satellite nucleic acids. And then there's a special word called virusoids. Virusoids are circular RNA satellites. So basically you have satellites here. You have satellite nucleic acids. You have satellite, what was the other one? Satellite viruses. And a special kind of satellite viruses is virusoids. And what's different? They have circular RNA. Just a special name. What's the difference between a satellite nucleic acid and a satellite virus? A satellite virus has all its pieces. They just happen to be associated. A satellite nucleic acid, one of the genes for the protein coat in this one is located in that one. Okay? So, satellites are very strange. Notice their size. 500 to 2,000 nucleotides. What were viroids? Notice that they are 246 to 399. That makes them, test question, the smallest entity of disease. Smallest entity of disease on Earth is viroids. 246 to 399 RNA bases. Satellites, remember, they are 500 to 2,000. Again, they're mostly RNA. There's a few DNA. All right, and there are, as far as we know, no human satellite viruses. There are a few, I guess you'd call it an animal because they are of insects, called chronic bee paralysis. That's the one animal satellite virus that we know of. And so it's just something for us to know about. You don't have to know hardly anything about satellite. So if you put what you got in your table, your data table, it cannot be wrong. If you lie, it's not only an F on it, it's a 0 on a required component of the class. What is lying or cheating on the paper? Saying we did a test we didn't do. That means you copied a paper from a previous term. Every term we do slightly different tests. So if you put in there lysine iron or peptone iron deeps, I'll know you either didn't know squat or you lied, both of which are 0s, because we didn't do that this term. If you say blah 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 test, I got a positive, remember there was one test that didn't work. I'm the only one that knows it. So if you got a positive, there's going to be some serious repercussions because it didn't work. The reagent was bad. So whatever you got is perfect. All right, when you go to the data books to fill out, remember you're supposed to go through 6, 7, 8, 9 plus systematic and McFadden or anything online and find something that most closely matches your results. So you're going to have a data table, and the first column is going to be every test we did, alphabetized, which is the same first column of the methods page. Then there's going to be a column, and this is going to be called tests, and the next column is going to be called you. Now don't put my name. Every term I have somebody put Hicks. Or if I change it to my name, they put my name. Put your last name. Because nowadays, they only record in the reference books the results that differentiate it from another genus. Not all the tests like we did. Finally, what if you got something no one else on earth has ever gotten? Like use on motility. And you got a positive and every reference book you checked said negative. Then you have to put an asterisk in front of motility, and down here you say this unusual and strange and twisted result. No, you don't have to say twisted or unusual. You can just say this unexpected result. It's explained in the third paragraph of the conclusion, which we call the excuse paragraph. How I could have possibly done this wrong. All right, so anyway, these are some of the hints. What do people do that cheat? They add tests we didn't do. They put a list of tests that we did and they make up results. Or they go to a reference book and it doesn't include that, so they just put something. All of which, remember, in 23 years, we've had the same 35 unknowns. I have memorized them all. I know what it should be. Don't lie, cheat, steal, or alter the results. Nothing you do from a reference book and nothing you did on a test can be wrong. All right, yeah.
We have repeated a test. Does that go in this data table? Twice?
Yeah, you could probably put like motility 1 this, motility 2 that, and you can even write inconclusive if you couldn't, and that's why you repeated it. Okay, so anyway, that's a little thing, a little more. Smith, McGillicuddy, Nguyen, whatever, put it in there. And then you should have a result for each one of the tests. These are the ones you did. If you didn't do that test, don't put a result. Just leave it out. And remember, there are several tests that have more than one result, like Ch10AViroidsPrions. It has whether or not you're a gram-negative on there. It has a color, how it grows on agar. If it's clear, it's lactose negative. If it's salmon, it's lactose positive. If it's green, it's Shigella. If it's blue-green, it's salmonella. Or no, just the opposite. But then, anyway. And then it has hydrogen sulfide crystals. So there are 3 things you should have recorded about Ch10AViroids. Litmus milk. There is no, don't be giving me positive on litmus milk. That's crap. There are two things on this milk, lactose and casein. On Kligger's iron, there are three things, glucose, lactose, and hydrogen sulfide, and you could even have a gas on glucose. So don't be telling me positive. That'll drive me nuts, because that means you didn't know squat. All right, so on the data page, you're going to have reference 1, and of course, you're not going to put reference 1. You're going to put like Bertie 6th, 7th, and 9th, or whatever, as the other columns. Remember, this one most closely matches your results than these. And the newer the book, the less results it quotes. It might have only 5 results. People made official Fs because they came here and they actually flunked it. But a total of 15 people got an F. And that counts the 5 people that dropped, and then the other people that just didn't bother to call me or show up. Of which 4 had a B, which still upsets me. Which, if you look at the night classes, the other night class, it's pretty favorable comparison, similar. Actually better than usual for my midterm. My midterm usually averages 67. So
What was the average?
I haven't gotten it completed yet because I want to do these 2. I was up till 4 last night trying to get it all done. Anyway, so that means that everybody's going to be getting a midterm update grade sheet, and there will be another set of bonus questions available for everybody in the class if they have them done by the 22nd. So we're going to talk about that. Friday is how to type a perfect unknown report, and yes, I'm going to go a little bit over it next, but if you want to know all the details and you want to look at a couple of them that I don't have online, you can certainly come, and I will be talking about that, and it would be very nice if you have questions. So you should look into how to do it before you show up. So if you have questions, then we will be able to answer them for you. And remember, there is a couple of things that I want to make sure people know. Whatever data you got is right. You cannot collect data that's wrong. Very pretty picture for anybody. All right, so anyway, this is the first class after the midterm. And I'm going to show you where a few things are and discuss a few things about what's here. First of all, after the curve, and I'm not even going to tell you what the curve was, but after the curve, 7 people made an A in here, and 4 of them got 100%. Most of that 100% came from the people who absolutely kicked butt on the word questions, because there was 80 possible points on the word question.
80? Oh,
80.
One of them is 2.
Yes, 2 of them.
2 of them.
2 of them.
So that was great. A huge number of people, which means you know I'm going to do it again, did not read the directions. And on the directions of the exam, it says, if you read these directions, record the answer to number 206 as mycobacteria. 10 points free, 20 multiple choice questions. If you don't learn to read the instructions, I am going to keep doing it. You know, if it says when you're turning on some sort of respirator at the hospital and you don't read the instructions and it's not the model you were trained on and you set it up wrong, somebody's going to die. Read the instructions, always. So anyway, in your class, 7 people make A's, 2 people made B's, 6 people made C's, 5 people made D's, 6